Hello, welcome to Chapter 4. This chapter from your textbook is all about the very broad field of molecular biology. And what is molecular biology? Well, there are several subjects or topics, I guess, or fields of study that would fall within the category of molecular biology. You're dealing with genetics, genes, DNA, RNA, protein synthesis. We will also talk about how cells divide in part 2 of this. Now, of course, your textbook presents a great deal of information about this, but I have done my best to narrow it down to just what you need in this class. You can take a whole entire course just on molecular biology. In fact, people get PhDs in molecular biology. That means all they do is study this. All right, so just to let you know, it's a huge subject. So like I said, I've done my best to narrow this down just to what this particular class really needs. And really in most biology classes, you should cover some aspects of DNA and RNA and genetics. All right, so again, I'm keeping this as basic as possible with just the highlights. So let's get started. So as I sort of hinted, you do need to have some familiarity with, hold on a second, that's what I want. You do need to have some familiarity with DNA, RNA, and genes. For our purposes, when we talk about diseases, many of the diseases that you have heard of or will learn about are genetically based. Okay, we can reduce the cause of that disease to some sort of problem with DNA, genes, or proteins. And so that's really why it's so important for us to have some idea of what DNA and RNA are all about. Some examples of diseases that are related to some sort of DNA mutation, for example, or some issue with forming the proper protein. And these are just a handful of examples. There are really so many diseases that can be traced back to some error in DNA synthesis, for example, or some problem in a genetic sequence. So again, these are just some examples. Color blindness, cystic fibrosis, diabetes, hemophilia. All right, these are just some examples of diseases that are related to some, mistake or error in DNA synthesis, DNA replication, or something along those lines. For the rest of this slide, I don't really care that like I'm not going to ask you questions about any of this except for maybe this point that's underlined. You should basically know that it was in 1953 that the overall structure of DNA was discovered. Now, earlier than that, actually by 1900, the components that make up the DNA molecule were discovered. And as we'll see, what makes up a DNA molecule? Well, you're going to have some kind of a sugar, you're going to have phosphate group, and you're going to have a nitrogenous base. And we're going to go over all three of these, and I will ask that you do know what makes up the DNA molecule. And like I said, we're going to go over it here in a minute. But as far as this slide goes, I think, really the big important fact here is when, you should know when DNA was discovered, the structure of it. And it really wasn't that long ago. 1953 to some of you might sound like a long time ago, but it really wasn't. So it's still kind of new to us, and we're still figuring out new facts about DNA and RNA and genes as we go along. All right, so we still have a lot to learn about it. And then just kind of an interesting point, I think everyone will, point this out in an introduction to DNA, that in 1962, there were three men, last name Watson, Crick, and Wilkins, that were given the Nobel Prize for the discovery of DNA. But really, Rosalind Franklin was another scientist that was working with these guys, and she never did receive any recognition for her role in that. So good to keep in mind. It wasn't just these three guys, but also Rosalind Franklin. Much later, she started to get credit for it after she passed away. So anyway, there's that. So let's get into the DNA structure and function. DNA, what does it stand for? Deoxyribonucleic acid. Deoxyribonucleic acid. And so DNA is actually a very long molecule, and it actually will vary in length, all right, depending. But for everyone, we have 46 DNA molecules that are going to exist in the nucleus. Very important that you know that we keep DNA in the nucleus because we have to protect this very important molecule because this DNA contains all of our genetic information. So it's really important to protect this DNA from getting damaged. All right. It also, keeping it in the nucleus also helps protect DNA from outside influences such as maybe a bacterium or some sort of virus, especially viruses, because viruses really like to get to your DNA. But so, you know, it's important to keep it protected as much as possible. So DNA will not leave the nucleus. It will stay in there. As far as these numbers, like, you know, you don't have to know that it's 2 inches on average long. I won't ask you that. You should definitely know, though, that we have 46 DNA molecules living in the nucleus. Now, DNA, and there are other nucleic acids. DNA is categorized as a nucleic acid. Now, DNA and other nucleic acids are polymers of nucleotides. So you get a bunch of nucleotides together and they will form DNA. All right, so we're going to break it down and look at what is the structure of a single nucleotide. All right, remember, nucleotides are going to be monomers or individual units that when they bind together, they're going to form this larger polymer known as DNA. So one individual nucleotide, we need to go over the structure of this. This is definitely something you'll be asked about. So one nucleotide is going to consist of a sugar, a phosphate group, and a nitrogenous base. Nitrogenous base, this means that this chemical structure called a base is largely composed of nitrogen. So that's why it's called a nitrogenous base. So let's look at this. So in words, here's your description of what a nucleotide is composed of. As I mentioned, you're going to have one sugar, one phosphate group, and one nitrogenous base. Now, when we're talking about DNA, the nucleotides that make up the DNA molecule, the sugar that is making this up is called deoxyribose. There will be one phosphate group. There will be one nitrogenous base. Now, the nitrogenous base can be one of 4, okay, different types. So the nitrogenous bases are classified into 2 different groups, first of all. You have purines and pyrimidines. Purines, there are 2 nitrogenous bases that are classified as purines. We have adenine and guanine. For adenine, we just use the letter A. For guanine, we just use the letter G. The second class of nitrogenous base is called pyrimidine. Okay, so there are actually 3 types of pyrimidines. You have cytosine, thymine, and uracil. Now, uracil is not present in the DNA molecule. Uracil, as we'll see, is present in the RNA molecule, not DNA. Okay, so for DNA, what you find as representing one of your nitrogenous bases that make up the nucleotides, that make up the DNA molecule, are going to be of 4 types: adenine, guanine, cytosine, thymine. You must know those 4. And you'll need to know uracil as well. But again, that's relating to RNA. Right now, we're just going to deal with DNA. So yeah, the DNA bases, the DNA nitrogenous bases are going to be one of these 4. okay, for each nucleotide. You have adenine, thymine, cytosine, guanine. Now for a look at the structure of this, okay? So remember, again, we're saying that nucleotides are the monomers that make up the DNA molecule, okay? So a single nucleotide is going to look like this chemically. As I mentioned, you're going to have a sugar group, which if we're talking about DNA, that sugar group is called deoxyribose. All right, and again, you know, I'm weird about using the word sugar. This is a carbohydrate. I actually prefer that term, but sometimes I may say sugar. All right, so deoxyribose, this is your carbohydrate or sugar. All right, and then you're going to have a phosphate group. All right, so the phosphate group, you've heard of phosphates before. We've talked about phosphate in relation to ATP. Now, I'm not going to ask that you know the chemical structure. You don't have to memorize the chemical structure exactly, but definitely you do want to know what these nucleotides are made of. So we're going to have a deoxyribose, we're going to have a phosphate, and then up here is our nitrogenous base. This particular example is showing you adenine. Okay, remember, we have 4 different nitrogenous bases that can attach to the deoxyribose here. And that could be adenine, as shown here, or it could be guanine, it could be cytosine, or thymine. And those will all have slightly different chemical structures. All right, but as you can see, there are a number of nitrogens, okay, and for nitrogen, there are a number of those here. So that's why it's called a nitrogenous base. All right, so definitely you want to know what is going to compose a nucleotide. Here are just some other examples of the other nitrogenous bases. Again, don't try to you know, memorize the exact chemical structure of these. You do not need to do that for this class. If you ever take a biochemistry class and maybe some other chemistry courses, maybe like organic chemistry, a few different types of chemistry classes, you will have to memorize the structures of these nitrogenous bases. And you know, all of DNA and RNA. That's a big topic in many of the chemistry courses. So if you ever do take a chemistry course, you probably will have to memorize these and be able to write these out. Okay, but you won't have to in this class. This is just showing you adenine, guanine, which are classified as purines. Purines are going to have a double ring. Okay, a double ring in the chemical structure, and then you have pyrimidines, which will be a single ring, and those are going to be cytosine, thymine, and then uracil when we're dealing with RNA. More on the DNA structure, the structure of this molecule. You might remember, because we talked about this in chapter 2, a little bit about DNA. You might remember that DNA takes on a helix shape, a double helix shape, and it kind of looks like a spiral staircase. DNA is double-stranded, and so those 2 strands will sort of coil and give it this helical shape. And we're going to see a picture of this here in a minute. Each of the 2 strands, remember DNA is double-stranded, each of the 2 strands serves as like a backbone of phosphate groups, which are alternating with the deoxyribose. And don't worry, like I said, we're going to see a picture of this in a minute. It makes more sense when you get a visual. And then the nitrogenous bases are going to extend from each strand, and they're going to pair up, okay, in a specific way through something known as base pairing. And you've already learned a little bit about base pairing in chapter 2, right? We talked about a little bit about what are the nitrogenous bases making up DNA. We already mentioned adenine, cytosine, thymine, guanine, that these are the 4 bases and that they pair up in a very specific way. So that thymine pairs up with adenine and cytosine pairs up with adenine, all right? So the nitrogenous bases, and again, as we're going to see here in a picture of this, nitrogenous bases are going to bond together by way of hydrogen bonds, okay? Hydrogen bonds we haven't really talked too much about, but it's just what it sounds like, all right? Two hydrogens are going to bond together. They're going to form a chemical bond together. And so in this way, that's how the pairs of bases, all right, like T with A or C with G. I think I misspoke on the last slide. I think I misspoke on a pairing. All right, this is the deal. Adenine and thymine pair together. Cytosine and guanine pair together. I think I said something different on that last slide. I'm talking too fast. All right, so here it is, right? Adenine pairs with thymine, cytosine pairs with guanine, and they're going to pair together by way of hydrogen bonds. All right, so for example, a hydrogen on adenine will pair up with a hydrogen on thymine. Hydrogen on cytosine will pair up with a hydrogen attached to guanine. So adenine and thymine pair together by way of 2 hydrogen bonds. Cytosine and guanine pair together by way of 3 hydrogen bonds. And what happens here is that it's a purine bonding together with a pyrimidine. And again, here's a reminder of your DNA base pairing. And essentially this makes up the entire genetic code. And this relates to a law called the complementary base pairing law. And this is just that when one strand of DNA pairs with the other strand of DNA, remember DNA is 2 strands, and those 2 strands are going to chemically bond together by way of this base pairing. Okay, so in essence, one strand ends up determining the base sequences that will exist on the second strand. And again, don't worry, it's going to make more sense when we look at the picture here in a second. And here is the DNA structure. This is a slide, especially this image on the right-hand side, that I do recommend taking a look at this repeatedly, because this gives you a good understanding of how this DNA molecule is structured. So this blue strand in the back, okay, so you've got 2 strands of DNA. Remember, it's double-stranded. So you can see one strand here on the left-hand side, and then there's another strand here on the right. So what makes up these strands? Well, the little parts in green, all right, those are the deoxyribose groups, all right, so those are the carbohydrate groups or the sugar groups. Okay, so the ones in green, these are deoxyribose. Attached to each deoxyribose is a phosphate group. So that is shown here in yellow, the little yellow circles. All right, so basically the backbone of each strand is going to be composed of one deoxyribose with a phosphate attached to it. Okay, and you can see that actually it's the phosphate groups that are attaching the deoxyribose in this linear sort of fashion. All right, so you should definitely know that the deoxyribose groups, along with the phosphate groups attached to them, these are what are making up the backbone of the actual strands of DNA. Okay, so you can see that on the left strand. You can see the same thing on the right strand. Now, how do these strands end up being connected or linked together? They have to be linked together. So that's where the nitrogenous bases come into play. So let's look at this first strand here on the left. This particular deoxyribose here has attached to it the base or the nitrogenous base known as adenine. Down here, this one is associated with the nitrogenous base known as guanine. This one here has thymine on it. This one down here has guanine, so you can see that. What's going to happen is this adenine over here on the left, this adenine is going to demonstrate what we call base pairing. So the adenine is going to pair up with thymine on this side. So in essence, this one strand will determine the sequence, the base sequence of the other strand. All right, and so this is just going to be the pattern all along this really long DNA molecule. All right, notice guanine over here is going to bind with the cytosine over on the other side. G goes with C, A goes with T, right? And all along, this is the pattern. And what ends up happening after this base pairing occurs, what ends up happening is the DNA molecule will take on this helix-like shape. It will just sort of coil around. All right? So please do take a look at this picture, a few times, enough so that you understand how this structure is. DNA structure and function. So, well, let's define what a gene is. Now, people will give you different definitions of what is a gene, but in general, you can consider a gene to be defined as a part or a segment of DNA that codes for the synthesis of a certain protein. Students often find it surprising that the genetic code, or what sequences we see on DNA, are actually a code for the synthesis of a certain protein. Like, why would all of our genetic information be all about making proteins? Well, that's because proteins are what make us function. So many of the processes that are going on in the human body are caused by the action of some kind of a protein. Okay, it's really all about proteins. So yeah, you can pretty much deduce your genetic code down to codes that are specific for making a certain protein. And we're going to talk about that. What is the genome? The genome is simply referring to all of the genes in one person. I'm not going to ask you these numbers. All right, this is just there if you're interested. All right, humans have about 20,000 genes, and this is going to equate to about 3.1 or so billion nucleotide pairs. Remember that DNA structure we looked at? 3 billion. All right, genomics is simply the study of the genome. Now, I do want to just point out that DNA has segments that do code for a specific protein, right? As represented by the definition of a gene. But you should keep in mind that there are also segments of DNA that are called non-coding DNA, and that's just what it sounds like. There are segments of DNA that do not code for anything that we really know. And that is actually being studied very intensely. Is, well, what are these segments of DNA, or what are these genes that don't code for anything? It may be that it does code for something, we just haven't figured out what that is yet. That could be it. All right. Other people have referred to some of this non-coding DNA as junk DNA. And then there are a lot of theories on, well, what is junk DNA? Just junk, like it's useless, it's there for no reason. Well, but the thing is, why would it be there for no reason? There's a reason for everything in the human body. So some people do believe that the non-coding or some of the junk DNA is maybe representing segments of DNA or genes that were once beneficial to human beings but are no longer useful. And it's just sort of like genes that are on their way of being phased out of the genome. Anyway, I could go on a tangent with that. There are lots of theories on what this is all about. What is this DNA that is non-coding or doesn't code for anything? And again, as we go on in life, scientists are finding more and more that these different segments of non-coding DNA are actually coding for something. Okay, so again, it could just be that some of these genes, we just don't know what they're coding for yet. All right, so chromatin and chromosomes. What is chromatin? Chromatin is basically condensed DNA. Remember I mentioned that DNA resides in the nucleus, but I also said that DNA is a really long molecule. So how do you fit such a long molecule into such an extremely tiny space, the nucleus? Well, you have to have DNA become condensed. It has to exist in a smaller form, a form where it's not taking up as much space. This is how we're able to fit DNA into the nucleus, is that DNA will become condensed. When it is in its condensed form, we call it chromatin. Okay, so I'm going to go over a little bit as well about how this DNA is complexed with proteins. Now also, chromatin will condense. Okay, so we're going to make it even smaller. Chromatin will condense. When chromatin is condensed, we call this a chromosome. Okay, so essentially, a chromosome is very, very condensed DNA. And so just so you know, like from smallest to largest, what we're dealing with here. First you have DNA, and that will start to condense into what we call chromatin. And then chromatin will become condensed even further, and then we have what you call a chromosome. In the nucleus of most cells, pretty much every cell of the human body, except for cells that you find in the ovaries, cells that you find in the testes, okay? But outside of those cells, all the rest of your cells in the body are going to have 46 chromosomes residing in the nucleus. 23 from each parent. All right, so half of these chromosomes come from mom, the other half come from dad. And again, you know, DNA, it's a long molecule. It's long, all right? You could actually stretch it out this long, like 6 feet. So obviously, you have to be able to make this DNA take up a much smaller space in order for it to be stored in the nucleus where it can be safe. All right, and some of these other points are just there again for your, you know, if you're interested. Here's some detail about chromatin structure. This is a really good slide. I love this slide because it is step by step telling you about how we're going to condense this DNA molecule. So let's start at the top. First, you have your DNA double helix molecule. You can clearly see that here at the top. That's going to start coiling more and more and more. And then what ends up happening is that this DNA molecule will actually wrap around something called a core particle. What is a core particle? A core particle is made of 8 histone proteins. This is mentioned again on the next slide, so don't worry. So basically, remember, I said that DNA is going to wrap around or condense around protein. Well, the name of that protein is called histones. And the histones, there will be 8 histone proteins together. And once those 8 histones are together, we then call that a core particle, a core particle. So the DNA is going to wrap around the core particle and stay stuck to it. And then these different core particles, which are made of histones, are going to be linked together by way of what's known as linker DNA. So basically the linker DNA is a specific segment of DNA that codes for just serving as linker DNA. Linker means we're linking together the core particles. Okay, so we're going to link all the core particles together by way of this linker DNA. Now, when you have a linker DNA together with the core particle, we call that something else. We call that a nucleosome. Yeah, there's quite a bit of terminology in this chapter. All right, so there's that part. All right, and then you're just going to see a whole bunch of nucleosomes, and see how we're packing this all together really tightly. All right, and then the nucleosomes will start to coil, and then you end up condensing further and further. You can see by these numbers how we go from larger to smaller. All right, we're going from millimeters. We're going to go to nanometers. All right, ultimately what we get is a chromosome. All right, we're going to form a chromosome, which is really, really condensed chromatin. Chromatin is really, really condensed DNA. Now, when we have a chromosome, there are certain parts of the chromosome that we're going to label. So basically in the middle, all right, in the middle of this chromosome, you have what you call centromere. And this is just like a pinching in of the two chromosomes. All right, so we do have, it's a pinching in of the chromatids. Okay, I failed to mention that chromosomes are actually made of chromatids. You're going to have two chromatids. They're called sister chromatids. And they're going to pair together like this. And they'll sort of be held together here in the middle by what you call a centromere. And I do have another slide explaining that later on. Here's in words basically what I just showed you on that previous picture. Histones. Histones are the proteins that DNA will wrap around. Okay, remember, histones are going to cluster together in groups of 8. Once we have those 8 histones bound together in a group, then the DNA molecule will end up wrapping around those histones. Okay, it will wind around those histones, sticking to it. And then you get the formation of a nucleosome. And what is a nucleosome? A nucleosome consists of a core particle. And remember, what is a core particle? A core particle is simply the cluster of 8 histones with DNA wrapped around it. Okay, that's a core particle. And then you have your linker DNA, which is just a short sequence or a short segment of DNA that just serves to connect the core particle together. So when we have our core particle and linker DNA, you can then call this a nucleosome. Okay, and so as you saw in the previous slide, nucleosomes are going to just sort of zigzag along, okay, sort of that kind of arrangement, and you will see all this looping and coiling going on, and ultimately you end up with a structure that is significantly shorter in space. It takes up much less space than the original DNA molecule. Now, as we're going to see when we do part 2 of chapter 4, we're going to talk about how cells divide. All right, so we'll talk about that later. All right, but when the cells do divide, what we need is to make a copy of DNA. We basically need to replicate or copy DNA to form new chromosomes, because we'll need that DNA to go into the new cells. All right, so again, we'll leave that conversation for part 2 of this. Now, as I was trying to show you earlier, each chromosome is going to consist of 2 chromatids. And so these 2 chromatids do have identical DNA. All right, but when we do cell division, what we're going to end up doing is splitting that chromosome apart so that we have our 2 sister chromatids separated from each other. All right, but for now, you can just understand that the chromosome is made of chromatids. You're going to have two, all right? And we just call them sister chromatids. And chromatids, as I pointed out, are joined at the center at an area called the centromere. Now, another structure associated with chromosomes is something called kinetochores. Kinetochores are going to be little proteins that are situated basically on the lateral aspects of the chromosome. And these are going to be important, again, later when we talk about cell division, that these kinetochores are going to help serve as attachment sites for microtubules in mitosis. Hopefully the word microtubules sounds familiar to you. Again, don't worry about it so much right now. We'll deal with that when we do part 2 of this. Okay, but for now you can just know the name kinetochores, and that these are protein plaques or protein complexes that exist on like the lateral sides of each chromosome. And here's a picture of that. So you've got your whole entire chromosome here. And what you're seeing are two halves. You're seeing that there's basically two parts of this chromosome. You have one sister chromatid and another sister chromatid. Okay, the two making up the entire chromosome are called chromatids. And if you look at this picture, it is showing like a coiled up structure, right? This is all chromatin. Remember, chromatin is super condensed DNA. Okay, so that's what they're trying to show here, all this coiling. The middle of the chromosome is referred to as the centromere. Centro, like center, so that should be easy to remember. It's the center. And then on the lateral aspects of this chromosome, you see these other proteins. These are the kinetochores. And the kinetochores, again, we'll bring those up when we talk about cell division in part 2 and what role these play. So you should definitely be sure to know your chromosome structure, how that looks in those different parts of the chromosome. You should know what sister chromatids are. You should know what a centromere is and what the kinetochores are. So that was DNA. Let's talk about RNA. What is RNA? RNA, like DNA, is a nucleic acid. RNA, also like DNA, will be composed of nucleotides. Now, RNA is a little bit smaller, okay, than DNA, but it does resemble DNA a lot. Why is that? Because RNA is essentially going to serve as a copy of DNA. Now, there are 3 important types of RNAs that are going to be associated with the very important process of protein synthesis. 3 types that we will mention, and you need to know all 3 of these. We have messenger RNA. You can abbreviate that as mRNA, messenger RNA. Messenger RNA is probably the one that gets the most attention in research. Any scientist or molecular biologist who is studying RNA, typically they're going to put a lot of emphasis on their study and understanding of mRNA. Messenger RNA, like I said, gets a lot of attention. That's because messenger RNA is a copy of DNA. It's a copy of the DNA strand. And so messenger RNA is just what it sounds like. Messenger RNA is going to go and get a copy of DNA in the nucleus. Okay, remember, DNA lives in the nucleus. It does not leave the nucleus. Messenger RNA, however, can go into the nucleus and come back out into the cytoplasm. So messenger RNA goes into the nucleus. It's going to copy, okay, there's going to be a copy made of DNA. And then that's actually when we really call it messenger RNA. Because at that point, this molecule is serving as a messenger to the rest of the cell about what the DNA code is. So messenger RNA is a copy of DNA. And then RNA is going to leave the nucleus, right? And then carry that message out to other areas of the cell. Okay, so messenger RNA is really cool because it can go into the nucleus, get your DNA copy, okay, and then leave the nucleus, right? Because, you know, DNA can't leave the nucleus. So it's really a big deal, this messenger RNA, because this is the molecule that's going into the nucleus, getting the genetic code, and then leaving the nucleus and giving that message, giving that DNA code message to the rest of the cell. All right, so another type of RNA is called ribosomal RNA, or rRNA. Ribosomal RNA, you might conclude, is going to be associated with ribosomes. You remember ribosomes. Ribosomes are the site of protein synthesis, right? Remember, you have ribosomes that exist on the surface of the rough endoplasmic reticulum. You also have ribosomes that are considered free-floating. These are ribosomes that are not attached to the rough ER, and they're just going to be out in the cytoplasm. Okay, so this rRNA is associated with those ribosomes. Okay, and the third type of RNA is called transfer RNA, or tRNA. Transfer RNA is going to play a role in some of the later stages of coding for a protein. Basically, and we're going to see that transfer RNA on one end of the molecule is going to be attached to an amino acid. And then the other end of that transfer RNA is going to be associating at the ribosomes. And then ribosomal RNA is going to help, along with transfer RNA, basically convert the language of molecules of DNA and RNA into the language of proteins. Okay, so we're going to transfer, okay, this language, okay, and we're going to see some pictures of that, so don't worry. What else about RNA? You might remember from chapter 2 that RNA is a single-stranded molecule, single strand, not a double strand like DNA. And another difference between DNA and RNA is that the carbohydrate group, or the sugar group making up RNA, is called ribose, ribonucleic acid. And then you might also remember that for RNA, remember, both DNA and RNA are going to be associated with 4 nitrogenous bases. And DNA, one of those is thymine. Well, in RNA, we don't have thymine associated with RNA. We have a different one in place of that. And in RNA, we have uracil instead of thymine. So you should definitely know that, you know, when we're dealing with RNA, we don't have thymine. Instead, we have uracil. Remember, another difference here, and I think I already emphasized this, is that RNA is mainly going to be functioning in the cytoplasm, whereas DNA does not go into the cytoplasm. All right, DNA is going to stay in the nucleus. RNA is mainly functioning in the cytoplasm, but as mentioned, RNA can and will go into the nucleus, all right, just to copy the DNA, and then it's going to come right back out to the cytoplasm and do all kinds of things. I think I already recorded this slide, but it didn't actually record, so forgive me if I go over this slide twice. So what is a gene, right? We already said that a gene is a segment of DNA that carries a particular code for a particular protein. So it really all comes down to the pairing between the nitrogenous bases. A with T, T with A, right? G with C, C with G. What protein you end up getting is based on the sequence of these base pairings all along the whole length of the DNA molecule. It really all just comes down to those 4 nitrogenous bases, ATGC, See, and the different combination that you will have of those pairings throughout the length of the DNA molecule. And remember what this is all for. The DNA code, the nucleotide pairings and the sequence is all for one thing. It's all to tell the cell what kind of protein to make at any given time. It's all about that. The second point here is really just saying the same thing but differently. Remember, proteins are made of amino acids. Amino acids are the building blocks of proteins. So essentially what's happening is that the DNA molecule and the nucleotides and the pairing of the bases are what predict the specific primary sequence of a protein. Okay, you remember how a protein develops, right? We have a primary, secondary, tertiary, and quaternary stage of development for proteins. So that first stage is the primary stage or the primary level. And that is just referring to the specific amino acid sequence that will determine what that protein is. Okay, so yeah, the genes are telling the cell what amino acid sequence should be happening at any given time. And again, this comes down to the nucleotide sequence in DNA. Now, the rest of this information here, I'm not going to ask you about any of these numbers. That's just there for your interest. Genomic medicine, I think I already hinted about this. Yeah, I mean, it's important to have some knowledge about the genome. And this is really important for real-life applications so that we can figure out, if we know the specific cause of disorders and disease, then that can help us predict whether or not an individual is going to develop those diseases if they are genetically based. It's going to help us with diagnosis, right? If we can do like a genetic workup on a person, then we can diagnose that this person is definitely has a certain disorder, or if they might have a predisposition or a likelihood for getting that disorder in the future. And of course, at the end of the day, that helps us know, what to do to treat the disease, if we have a treatment for it. And again, knowledge of the genome and genes is going to be relevant to many different types of disorders that have some sort of genetic link. Alzheimer's disease, there is some genetic basis for that. I think it's only like 50 or maybe 50, 40 something percent. So like if a parent has Alzheimer's, maybe their child has like a 40 to 50% chance of also getting it. Schizophrenia, similar situation on that. Obesity, there is some evidence to suggest that there is a genetic component to obesity. AIDS, I don't really, I mean, I would say with that, it's just like if you have, if you're like pregnant and you have a baby, then probably that baby's going to get AIDS. But as far as the genetic basis for this, that's really all there is to it as far as I know. Tuberculosis, lots of diseases. Cancer is, and it doesn't matter what cancer it is, every cancer, the cause of it, the immediate cause of it, is cells dividing out of control. And we're going to mention a little bit about that in part 2, okay? The question is always, what caused those cells to start dividing out of control, okay? So cancer is related to the genome only in, or not, I shouldn't say only, but mainly in the sense of the fact that cancer is caused by over dividing. Too many cells are dividing, and you end up with tumors. Anyway, we'll revisit that. Okay, so main general point here is genomic medicine is important for these things. Okay, it allows us possible early detection of diseases. And, of course, continued study of the genome might allow us to do certain things like gene substitution, where basically we can splice out a bad gene and insert the good gene. The genetic code. So the genetic code is a combination of these four nitrogenous bases making up the nucleotides: adenine, thymine, cytosine, guanine for DNA. All right, the genetic code, I already mentioned what this is. All right, this is just where we have these four nitrogenous bases here that make up the four nucleotides that are going to code for a specific amino acid sequence, which will result in the development of a protein. Now, there's something called a base triplet as part of DNA. So what is a base triplet? Well, you already know what a base is. A nitrogenous base is going to be one of these four. Triplet just means three. Okay, so basically what we can do with the DNA molecule is we can divide it into what we call base triplets. And so this would be just a group of three bases. And what has been found is that basically in DNA, it is these base triplets that will code for a single amino acid. Okay, so this is how we're getting this code or this language, all right, from nitrogenous bases to protein eventually, is that base triplets are associated with specific amino acids. All right, so for example, the base triplet adenine, guanine, cytosine, those three together next to each other in a sequence in the DNA molecule will code for the specific amino acid known as serine. And then, you know, this will go on and on and on. Maybe there will be other triplets, maybe not AGC, maybe AUG or CTA or whatever, that will code for other amino acids besides serine. And then all these amino acids will link together by way of peptide bonds, which you learned about in chapter 2. And then eventually you get this growing chain of amino acids that will ultimately form a fully developed protein. Okay, so DNA has these base triplets. Now, what about messenger RNA? Remember, I said RNA is going to go into the nucleus and copy the DNA molecule. So when it does this, right, RNA copies all these base triplets. When that base triplet is on or has been copied to mRNA, we now call this a codon. All right, we call this a codon. And this codon will ultimately be read by the ribosomes. Now, you don't have to know this number, okay, but just to give you an idea, there are 64 possible codons available to represent all 20 of the amino acids. So basically you can get, you know, a wide variety here of what individual amino acids will be linked together. Okay, again, don't worry about those numbers. Now we get into a special subject, transcription and translation. So when a gene is, it says here, when a gene is activated, that statement right there is a loaded statement. And chapter 4 does go into this, but I did leave this out because it's just it's a really big subject and it can get kind of complicated. And I have to narrow things down. All right, so when a gene is activated, okay, so the process of activating a gene is really complex and it's going to depend on a lot of different factors working together. We're not going into how a gene is activated. All right, many different factors involved. We can also have a gene inhibited, which is another whole story. Okay, so but just when a gene is activated, this is when we're going to see messenger RNA created. All right, so mRNA will be a strand, okay, that is created that is complementary to the original DNA sequence. And let me actually go back a slide, I think, or can I do that? Hold on. Okay, well, for some reason, it's not letting me. All right, that's okay. So mRNA is complementary to the original DNA sequence or original DNA gene. And then what's going to happen? After this mRNA copies the code from DNA in the nucleus, it's going to leave the nucleus and go out to the cytoplasm, where it is then going to start coding for specific amino acids using its codons. Here is what some people describe as the central dogma of molecular biology. And it's simply stating that this is the sequence. First, we have DNA. DNA gets converted, or the code of DNA gets converted into a messenger RNA molecule. And then mRNA goes out and will start signaling what kind of protein to make based on this DNA code. This is actually going to be broken down into two processes. One is called transcription, and the other one is called translation. And we will mention a little bit about each. For each, we are not going into a lot of detail. We're just keeping it very basic and very simple with this because, like I said before, you can study transcription translation like your whole life, right? You can get like a PhD in that. We're not going there, right? Not right now. So in transcription, this is the first process, and it really belongs right here where this arrow is. In transcription, this is when we're doing the copying. This is when mRNA is copying DNA. Transcription. So to transcribe something means to copy something. So that's what's happening here. All right, mRNA is copying DNA, and that is occurring in the nucleus. So you must know what these processes are and where they're taking place. That's a definite test question. So transcription, DNA is going to code for mRNA. In other words, mRNA is transcribing or copying DNA in the nucleus. Then mRNA, once it's fully made, will leave the nucleus, go to the cytoplasm, and that's really where this arrow is, translation. Translate, just what it sounds like. Here, mRNA is going to actually code for the protein. We're going to see the genetic code or the codon from mRNA be translated into the language of protein. All right, we're going to see how this code is going to be translated to the language of protein or amino acids. You must know this is occurring in the cytoplasm. Here's just a brief schematic that I drew of this, all right? Important to know, right? DNA to RNA to protein. Transcription is occurring here. This is where RNA is transcribing or copying DNA. It's happening in the nucleus. Translation is the process by which RNA is going to translate the genetic code into protein. And this is happening in the cytoplasm. All right, we're getting there. Only a few more slides to go. So let's talk about transcription. A little more detail about it. Again, what is it? It's the copying of the genetic instructions on DNA to a new mRNA molecule. There is a very important enzyme called RNA polymerase. You must know this enzyme. Now keep in mind, there are different kinds of RNA polymerase. You have RNA polymerase 1, RNA polymerase 2, etc. We're not going to know all the differences between all the different kinds of RNA polymerase enzymes. Just in general, all right, understand that the RNA polymerase, or the group of enzymes that are considered RNA polymerases, they are going to be the ones that will actually open up the DNA helix and read the bases from one of the strands of DNA. Okay, so RNA polymerase is really essential in transcription. It is necessary to do transcription because, first of all, RNA polymerase is going to unwind the DNA helix. It's going to open it up so that all the bases in the DNA molecule can be accessible, so that they can be read. So RNA polymerase, after it helps to unwind the DNA helix, it will actually create a nitrogenous base. And this is really cool how this happens. I'm going to show you a picture. Okay, so wherever RNA polymerase finds a cytosine on DNA, it is going to add a guanine base to the mRNA strand, which is right there. Okay, this is part of this complementary base pairing that we can see in another process called transcription. All right, again, we're just going to see how our nitrogenous bases pair up in a specific way. But in this case, okay, and we're actually having RNA polymerase add a new nitrogenous base to the new mRNA strand, which is close by. Wherever RNA polymerase encounters a guanine on the DNA molecule, right near it, it's going to add a cytosine to the mRNA, the newly growing mRNA molecule right beside it. Remember, cytosine pairs with guanine, thymine pairs with adenine, and so on. Now, here comes into play this nitrogenous base known as uracil. Remember that in the DNA molecule, adenine binds with thymine. But during transcription, we have our mRNA molecule there that's newly growing. It's continuing to develop as we go on with this. Well, remember that RNA does not contain thymine, right? Instead, it contains uracil. So that means wherever RNA polymerase finds an adenine on DNA, well, normally adenine binds with thymine on DNA, but in RNA there's no thymine. So adenine will then bind with a uracil, or actually RNA polymerase will create a uracil that can bind with that adenine on, or match up, I'm sorry, with that adenine on mRNA. So you see how we're getting a copy. We're getting an mRNA copy of the DNA code. All right? Here's a picture of this. So transcription, remember, is to copy something. In transcription, DNA is going to be copied into mRNA. RNA molecules are produced by copying this part of the DNA sequence into what is called a complementary RNA sequence, and this will be the mRNA molecule. So let's just look at this picture. In blue here, the blue strands, that is DNA. All right, that's your helical DNA. You can see the nitrogenous bases that previously paired up to form the DNA molecule. Right? You can see here this very first one, adenine on one strand paired up with thymine on the second strand. And so on and so on and so on. What will happen here during transcription is that this real important enzyme, RNA polymerase, and you can see that in like this aqua-like color, RNA polymerase is going to unwind, first of all, it will unwind this DNA helix, essentially separating the bonds existing between the bases on that DNA molecule. You can see that happening here. The DNA helix is unwinding. Next, RNA polymerase is going to read the DNA code. And whatever nitrogenous base it sees there, or whatever nucleotide, I should say, it sees there, all right, it's going to add the corresponding base to the newly growing RNA strand. The RNA strand is shown here in red. Okay, so you notice RNA is a single-stranded molecule. So it will have like one base each. These bases are not paired with anything else at this time. All right, but let's look here. So let's say RNA, your red strand here, RNA, it has a U on it, right? Or I should say this opposite. Look at the DNA. DNA has a thymine here, where my little red thing is, thymine. So RNA polymerase is going to add a uracil. See how this ends up happening is that the RNA molecule will have a corresponding code to that of DNA. If the DNA strand had a T on it, like right here where my little thing is, thymine, then RNA polymerase will add an adenine onto the RNA strand. If the DNA has a cytosine on it, RNA polymerase will put a guanine onto the RNA strand. Okay, so basically RNA polymerase will help construct the mRNA molecule. So that's basically transcription. Transcription is the copying of the DNA and the creation of a new mRNA molecule by RNA polymerase. Once that's done, we're then going to do translation. What is translation? We're converting the language of nucleotides into the language of amino acids. And so, again, what's going to happen here? Messenger RNA is going to carry the copied code of DNA that it got from the nucleus out to the cytoplasm to ribosomes. You then have these other RNA molecules called transfer RNA. Transfer RNA is a different RNA molecule that goes and attaches to specific amino acids, and it's going to carry those amino acids to the ribosomes. And it's the ribosomes that will start adding these amino acids to a growing protein. Now remember we mentioned that mRNA will have a codon. The codon of mRNA is basically a mirror image of what the DNA code was. Well, transfer RNA also has a similar code, but it's called an anticodon. Okay, and all this is the mirror reflection of the codon of mRNA. Ribosomes, right? So ribosomes, you remember, these are organelles, and they're going to read this message that messenger RNA is sending. You can find these free in the cytoplasm, or you can find them on the rough ER. Now, you can also have some on the nuclear envelope. We didn't really go much into that because really most of your ribosomes are going to be one of these two. They're going to be on the surface of the rough ER or free floating in the cytoplasm or cytosol. Yeah, we do have this other third type of RNA called ribosomal RNA. Okay, so just what it sounds like, ribosomal RNA is existing here in the ribosomes. That's why it's called ribosomal RNA. Okay, and so what's going to happen is ribosomal RNA will also be reading this message from RNA. Here is a graphic of what this might kind of look like in terms of what's what. If you look at the red line or the pink line, that is an mRNA molecule. And the little blue pieces, those are ribosomes. This looks like these would be like free-floating ribosomes. Now, what's going to happen is that this RNA molecule is actually being read by the ribosomes. The RNA strand literally goes through the ribosome. Or I should say that actually the ribosomes move. They move right along the RNA strand, reading the code of messenger RNA. And so as a result, we're going to have protein. Protein are these little green structures. These are growing peptide or protein, I should say these are growing primary sequences. So basically sequences of amino acids. All right, some are kind of short, some are a little bit longer than others. Those are a little bit more progressed. All right, so yeah, the ribosomes are going to read the mRNA strand. And from that, the ribosomes will start putting out or producing these chains of growing proteins, chains of amino acids. This picture looks a lot more complicated than it is. It's trying to show you translation, a very basic understanding of translation. Over here, this large structure is the nucleus. Remember that in the nucleus you have these little pores. These are called nuclear pores. RNA, right, mRNA, is going to, after it copies, after it's done transcription inside the nucleus, it's going to exit the nuclear pores. See, like this strand here. It's going to exit the nuclear pore, this mRNA. And then what happens is the mRNA is going to be read by a ribosome. Okay, the ribosomes are just going to move along this strand, reading it as it goes. And then as it goes, as it gets more code and more information from mRNA, it will start producing protein. Okay? Now here's a little tRNA molecule here. Transfer. So transfer RNA on one end of it is actually going to be carrying an amino acid. Okay, this is where we're getting the amino acids to start making the protein in the ribosome. Okay, transfer RNA. tRNA is going out and getting amino acids and transferring those amino acids to the ribosome so that the ribosome can link together whatever amino acids are needed to make whatever protein that mRNA is telling it to make. All right, here is a close-up of this. So down here you've got your mRNA, your messenger RNA, and it's going to have its codons. All right, so remember codons are the mirror image of the base triplets that were found in DNA. You have GG, guanine, guanine, guanine right here. You have adenine, cytosine, cytosine right here. All right, they exist in 3. And these triplets in mRNA are called codons. Okay, and so these codons, each codon is coding for one specific amino acid. So the mRNA is coming into, or I should say, the ribosome is reading the mRNA strands so that it knows what code, what amino acid should I be, or what amino acid should be attached here? What should be, what amino acids need to come together to form a specific protein? Meanwhile, you're going to have your transfer RNA. Let's look up to the top here. Transfer RNA is going to basically attach to a pool of amino acids. All right, there will be just a pool of individual amino acids out here. And so the tRNA will attach on one end to these amino acids. The tRNA then, okay, once it binds to an amino acid, it's going to come over here to the ribosome. Okay, the tRNA is going to deliver the amino acid it's carrying to the ribosome. This way, the ribosome has an amino acid to start building an amino acid sequence. All right. Okay, so understand that tRNA is just carrying individual amino acids to the ribosome, and then the ribosome is going to start attaching the different amino acids together, forming a chain, a long chain. As we go along, as the ribosome keeps going along this RNA strand, it is going to continue to make longer and longer chains of amino acids, eventually building a protein. All right, here is sort of the later stages of translation. All right, again, we've got our mRNA strand. We've got our little ribosomes all around reading, okay? The ribosome is just going to move along that RNA strand, reading the code mRNA has. And then you're going to have, remember the role of the tRNA, transfer RNA brings the amino acids to the ribosome, and then the ribosome starts adding those amino acids together to form a protein. And then once we have the primary sequence of that protein, then we're going to start building, or basically that primary sequence is going to start folding on each other, or folding sort of in on itself. And then you get, your different levels. You've got, remember, alpha helices, beta pleated sheets. Alpha helices and beta pleated sheets will start folding in on each other to go into the tertiary level of protein structure, and then you'll have your final protein. All right, and then after this, ribosomes will disassemble. All right, basically a ribosome has two parts to it. A ribosome has like a large subunit and a small subunit. And when we need that ribosome to read the RNA, the large subunit will combine with the smaller subunit so that the mRNA is between them. All right, and so when we're done with this, the ribosome will disassemble because we don't really need it anymore. Next, what's going to happen now? Well, we've got our protein, and now we need to make sure that protein is what it should be and it's formed properly. So the next thing that's going to happen, okay, is that the ribosome, remember, ribosomes are attached to the rough ER. This is showing you the rough endoplasmic reticulum here. Ribosomes are either going to be attached on the surface or free floating. All right, here it is showing you that this particular ribosome is here on the surface of this rough ER. Well, once that ribosome is finished with that playing its role in making that protein, the protein is then sent right to the adjacent rough ER. The rough ER will do some modification of this protein. Okay, it's going to do, maybe folding wasn't completely done. Maybe a tertiary level of protein development's going on here too. And, changes or whatever that need to be made to that protein. Okay, so rough ER really finishes the, let me say, the first draft of the protein. And then it will, basically the protein's going to go into a vesicle. It will be pinched off from the membrane of this organelle, the rough ER, and there will be protein inside of that vesicle. This vesicle is just for transport. We need to get this protein sent to another part of the cell. So we're going to encapsulate the protein into a vesicle so that it can be transported to the next place. Here, just again, is another look, very, very simplified look at transcription and translation. All right, so remember, for transcription, you've got your DNA strand. Now, at this point, now I forgot to mention that the DNA strand, it being two strands, one of those strands, once RNA polymerase comes along and unwinds the double helix, then one of the DNA strands serves as what you call a template. And that's the strand that's being copied. That's all that means. All right, so mRNA then is going to be created here as a mirror image copy of the DNA strand, right? Remember, RNA polymerase will add these specific nucleotides to the growing mRNA strand, and this gives you a codon, right? You'll get sets of 3. Let's say this codon is uracil guanine guanine. That set of 3 codes for a specific amino acid called tryptophan. You don't need to know the amino acids. All right, and once we have our codon established, then we'll do translation, where that codon of mRNA is going to be translated into the language of protein so that we get our tRNA bringing over a specific amino acid, and then we have our protein. Just sort of a comparison of transcription and translation. All right, remember transcription, we're copying DNA, forming an RNA molecule from that. It's in the nucleus, transcription. Translation is when we're synthesizing proteins using the message RNA or mRNA is carrying. Occurs in the cytoplasm. Okay. Here is a review of peptide formation. I'm not going to repeat this because you can just review this on your own. I've already explained this, okay? But here's just sort of a step-by-step, very general look at that whole process. Protein processing, I want to mention a little bit about this, and secretion of the protein. So protein synthesis is not finished yet. Where we left off, we're still not finished. To work, the protein must fold, all right? We have to get it into these higher level stages. Some proteins we're going to need to keep in the cell, all right? They're going to be used in the cytoplasm. Now, typically, proteins that are destined to stay inside of the cytoplasm are going to be made by the free ribosomes. Proteins that are destined to leave the cell to function elsewhere are typically going to be assembled on the ribosomes living on the rough ER. All right, and so this really is most of your proteins. Most of your proteins will be assembled this way. The ribosomes on the rough ER. And then these proteins, like I said, we're not done yet. These proteins are going to be sent to another really important organelle called the Golgi apparatus or the Golgi complex. Here's the sequence. In blue up here is the rough ER. Studded all over the rough ER, these little tiny dots, these are your ribosomes. Okay, so once we have our protein formed by, well, I should say once you have like your initial sequence, like the primary sequence of the protein formed by the ribosomes on the rough ER, that protein is going to, and it's not a full protein yet. It's in the primary stage, maybe secondary. And it's going to be packaged, all right? It will be released from the rough ER in a vesicle, a transport vesicle. That transport vesicle leaves the rough ER by way of what we call exocytosis. You remember that term, hopefully. And then that transport vesicle will come over to this organelle called the Golgi apparatus. That vesicle containing that immature protein is going to fuse with the membrane of the Golgi apparatus by way of endocytosis, which should also be familiar to you. In the Golgi, we're going to have further modification and refining of that protein. Okay, so Golgi does really the final additions or modifications to the protein. So by the time this protein is ready to leave the Golgi, it's fully formed. All right, it should be in its full, ready to go position. All right, so Golgi will then release that mature protein in a vesicle by way of exocytosis. And then, let's say, we're secreting this. Let's say this protein needs to go somewhere else. It doesn't need to stay in this cell. It needs to go somewhere else in the body. then we're going to have that vesicle containing that finished protein, and we're going to have it fuse with the plasma membrane of the cell, and then that protein will be released and transported somewhere outside of the body. Also, just notice here, the rough ER in this image is showing you that it's right next to the nucleus. This here in purple is the nucleus. And that is a really accurate depiction. In the cell, yeah, typically the rough ER is going to be actually continuous with the nucleus. The actual nuclear membrane will start to like fuse with the membrane of the rough ER, so that the rough ER is actually sort of continuous with the nucleus. This is for really, really good reason. This proximity of the rough ER to the nucleus is not an accident. This is an important arrangement because what's in the nucleus? DNA. DNA cannot come out of the nucleus. RNA is going to go into the nucleus and copy DNA. Well, this way, everything's right close together, right? Our RNA is going to leave the nucleus and go directly into being read by the ribosomes on the surface of that rough ER, right? Look how close proximity. That's not an accident. Okay, so yeah, this is an accurate representation. Typically, your rough ER is going to be continuous, actually, with the nucleus, or right near it. We have finally reached the last slide of this part 1. All right, and actually I already said all this. This is just in words what I just described on that last picture. Okay, so newly made protein coming from the rough ER will be sent to the Golgi. The Golgi apparatus or the Golgi complex will further modify that protein. It might add carbohydrate chains, making glycoproteins. Glyco just means carbohydrate or sugar. Okay, so we do have substances known as glycoproteins where they're a combination of carbohydrate and protein. Okay, so it may add carbohydrates to proteins. All right, a number of things are going to go on here in the Golgi, which is a whole other study itself. All right, but really cool. And this is the final area here in the Golgi where the protein is formed 100%, and then when it's finished, it will be sent out of the cell, or it will remain in the cell. All right, and that's going to end part 1 of this. So enjoy that, and if you are reading the textbook, please be careful because there's a lot of detail there that we're not using in this class. It's just too much. We don't have time for all that. All right, so please just limit yourself to what I talked about. Thanks, everybody. Have a great day.